Welcome back, guys. In this video tutorial we'll be talking about the sequencing method called Maxam-Gilbert method. So I'm using an image which is a beautiful illustration of how Maxam-Gilbert process works, and actually I am taking this from the biochemistry book, and I'll be referring to that, and I'll put the link in below for the understanding. So let's talk about the Maxam-Gilbert method of DNA synthesis and DNA sequencing. Maxam-Gilbert method is described based on the name and of the discoverer, who are the two scientists, Maxam and Gilbert. And the thing is, the sequencing method of Maxam-Gilbert based on the very basic understanding which is called the chemical sequencing, because these sequencing methods rely on the chemical synthesis process. Because the thing is, let's say this is the strand or the DNA sequence that we need to sequence, right? We don't know the sequence, we have a DNA strand, unknown sequence, and we can sequence that DNA by using some chemical termination processes. So let's talk about what we mean by that chemical processes in much more detail here. Let me break it down smaller. Yeah, okay, that will be good. Okay, so let's begin with the sample DNA. So we have the sample DNA, and what we prepare here, we need to prepare a homogeneous single-stranded DNA from that sample DNA, because sample DNA is double-stranded and we don't want double-stranded DNA for our reaction. We want homogeneous single-stranded DNA, and we are just preparing it from our double-stranded DNA. And preparation of a homogeneous single-stranded DNA is pretty easy, because it is a one-strand structure of that DNA, because we know DNA has a complementarity nature. That means if we have a one DNA strand structure, we can ultimately guess the other complementary DNA structure pretty easily based on the Watson-Crick pairing, that is A pairs with T and G pairs with C, right? So here what we can see, that this is the homogeneous strand that we get here, right? So once we get the homogeneous single-stranded DNA, then what we do, we add a phosphate group, and this phosphate is radio-labeled. This is radio-labeled. That's why it's called P32. It's a very common type of radioisotope for phosphorus that is widely used in biological processes. So we add it into the 5' phosphate of that DNA. So once we add that 5' phosphate, this 5' phosphate to this single-stranded homogeneous DNA that we just prepared, we are ready to go with our reaction. Now this is the preparatory phase for this Maxam-Gilbert reaction. Once we prepare our DNA by tagging it with radio-labeled phosphorus at the 5' end, now we start the major process for Maxam-Gilbert, and that is the chemical, remember, chemical termination process. Now that means what we have this sequence, and we need to find what this sequence is all about. Now remember, we don't know, actually in this case for our understanding we are drawing the sequence, we know the sequence, and I'm just showing you how to do that. But the thing is, actually in reality we never know this sequence, that's why we are sequencing it, right? So it will work by the formula that we are going to study now. Now let's say in Maxam-Gilbert process, the idea is to add certain chemical agents which will degrade and cleave certain bases out of the DNA. For example, if this is a single-stranded DNA, now if we add the chemical called DMS or dimethyl sulfate followed by piperidine, both are the chemical agents, if we add those two things one after another, it will cleave guanine out of this DNA strand. Okay, so that is called the G reaction, and that reaction should be carried on in one particular test tube, that is called the G test tube, right? Or test tube number 1. So let's say G test tube. In the G test tube we just add DMS followed by piperidine, and we know that whatever sequences are there in unknown DNA, it is going to cut guanine out of it. So if it is cleaving the guanine out, it will make break in the DNA, right? Because how many number of times the guanine is present, it will create that number of breaks. For example, in this case there are 2 guanines present, so they are going to produce 2 breaks in the DNA, ultimately leading to the generation of 3 strands, and that's what is going on here, right? So it is a test tube number 1 or guanine test tube. Now in the test tube number 2, we have added other chemicals which will cleave adenine out of the DNA, and some amount of guanine. I mean, whatever time they are getting adenine, they will cleave the DNA, but only in certain cases they can also cleave guanine out. That is called A plus G test tube, right? So that is called A plus G test tube there. In the A plus G test tube, we have that chemical. I'm not going to talk about the name of chemicals and how they degrade it, but the idea is to cleave a particular base out of the scenario. So in the first one we have guanine out, in the second one we have adenine plus guanine. It means adenine definitely, but they will cleave guanine occasionally. Okay, so if you look at here in this image, I mean, this is adenine placed at the terminal, so no way of cleaving it. Now another adenine at the bottom, so one cleavage, another here, so two. So due to the presence of adenine twice, actually thrice, but one in the terminal, so it won't cleave anything, it won't change anything, but presence of adenine here twice, so it will actually cleave the DNA 3 times. So we'll get 3 cleavages for adenine, and if there are guanine present 1 and 2 twice, so 2 cleavage for the guanine. So what we get here is that 5, so 3 for the adenine and 2 for the guanine, so 5 cleaved DNA strands, and that's what we are getting here in the second test tube. Now in the third test tube, we'll add certain chemical which will break down thymine out of the place. So it will break the thymine out of the place. So as the thymine is cleaved out, and occasionally it can also cleave out cysteine, so cytosine, so it will cleave thymine and cytosine. Keep on repeating cysteine there. So thymine and cytosine, these 2 bases are there. Now the chemical that we place, it will cleave thymine and cytosine out of the place, right? So here, as you can see here in this reaction, how many thymines are there? 1, 2, 3, 4. So as a result, what it will, the number of the cleaved DNA they are going to generate here due to the presence of thymine are 4, and for the presence of cytosine it will be 1, 2, so 2. So it will be ultimately 2, and so actually how many number of times it is going to cleave? As you can see here, it can cleave from this thymine, so it can generate 1 from this thymine, so 2 from this thymine, 3, 4, 5, 6 for the thymine itself. And for the cytosine, it can be twice. So ultimately 8 cleaved DNA can be produced from this. So how many number of combinations is possible? You can just tell it by looking at this DNA structure. So 8 times now. And the last one, we add certain chemical which is only cleaving the cytosine out of it, right? So cytosine will be cleaved. So the fourth test tube is cytosine test tube. So there are 4 test tube. The idea is we will be putting in each test tube certain chemical which are selectively cleaving a particular nucleotide out of the DNA, and as it is cleaving that nucleotide out of the DNA, ultimately DNA is broken down into fragments, and we'll get number of fragments from each test tube. Now, so once we produced all these different fragments from our DNA of interest, now this last thing is to run the electrophoresis and look at the data, because after running the electrophoresis we will go for the radioactivity, radiography. With the radiography we can tell the position of each bands. How? Because we just marked them with P32, because P32 is a radio labeled isotope. As a result of that, in audio radiogram we can see the presence of each bands, and easily we can interpret the result. So once we get it, once you understand this thing, so I need to rub this because yeah, so let's scroll it down a little bit. Here we go, and let's take another color. Let's take blue, let's take red. So here we go. So we have this 4 test tube. Remember, one is for entirely G, one is for A adenine plus guanine, another one is for the thymine plus cytosine, and the last one is for the cytosine itself. So 4 test tube destined for 4 different types of combinations. So now what we can look, after that we'll load the gel, right? Remember, in each test tube we can run only one well of the gel. We cannot mix them, because if we mix them it will completely destroy the experiment, right? So it's very, very important. So that's kind of disadvantage for this type of experiments, because we need to run 4 different setups continuously from the beginning of the experiment. So then we load them into the well. After loading them into the well, what we'll check, we do the electrophoresis. So after the electrophoresis we do the radiography. Autoradiography is done. Now radiography plate is there. Now from this plate we can identify certain things. Now how can we identify? Let's see here. So this is the band pattern that we form. Now remember, when in each tube we not only get the breakdown product, but also we can get some of the products, some of the DNA strands which are not broken. We also add that for a marker, so that we can understand that whether the process occurred correctly or not. So in all of this result on gel electrophoresis plate or radiogram plate, we can see the presence of one band common to all of them, and that is the total length of base pair. Remember, whatever we are talking about, the number of nucleotide in the base pair is 12. So it's a 12 nucleotide long sequence we need to deduct. It's very small, but for the experimental purpose and for the understanding, we are taking it very small. But actually in real life, this Maxam Gilbert sequencing used to produce 200 to 400 nucleotide sequence at the same time, and it can be done. But it is not enough, because for human genome it is a billion base pair long. So in that case we require something more advanced. That's why we move on towards the advanced techniques and also next generation sequencing techniques. So if you look at here, that in all of them we have the 12 nucleotide long, the single nucleotide DNA sequence that need to be sequenced. But rest of that band, so that band is common to all. I don't need to bother about that anymore. Now to find out the sequence. Now let's look at here. Now the 11, so here comes the length of the bases in this particular lane actually. So you can also run the ladder sequence there, so that you can find the reference for the length. So the length for the 11 number 11 nucleotides, we can have 2 bands here. We are having 2 bands, one for the C and one other for T plus C. That means here in this particular 11 stretch nucleotide sequence, we have when we terminate C we have the 11. When we terminate both T and C, we also have the 11 from the 12, right? Remember, 12 is the actual one, so we have 12 nucleotide long. Now we are getting 11, so 1 is released. So if we cleave C out intentionally, we get 11 nucleotide long. If we get T and C both out, still we get 11 nucleotide long. What does that mean? That means C is common for both of them. That means the nucleotide we cut out is definitely C, not T. Because if it is T, then it will reflect differently in the next tube. Try to understand. This is the most important part for interpreting the result. Because C is common in both the case, and if we cut C out, as a result in both the cases we get 11 nucleotide long, only one nucleotide out, though we treat it for T and C both, right? That means the first one will be C. Now if we look at here the next one, the 10 nucleotide long. Now we are going for 10 nucleotide long. In the 10 nucleotide long region, what we get here is one for C, one for T and C. So same thing again. So if we block C, if we cleave C out, we get 10 nucleotide long sequence. If we block T and C both out, we also get 10 nucleotide long. So again C is common for both of them, no other bands. That means again it will be C. The third one is the 9 base pair long. So now 9. So 9 base pair, we can find one at G, another one for A plus G. What does that mean? That means from the 12 nucleotide long DNA, if we cut the G out only, we get a 9 nucleotide base, right? If we cut for A and G both, still we are getting 9 nucleotide. That means again in both of these test tubes, the common element to be cut out is G, and the result is same. So the position is occupied by G, right? Now the 8th, so from 12 now we get 8 nucleotide long sequence. and the band, we get only one band at A plus G, right? That means we get, so if we add the chemical which will eliminate A and G out of the place, we get 8 nucleotide long, right? Now if we look at the G only, in the G there is no band. That means if it's supposed to be G, in that case we should get a band in the G, right? Because if we cleave this 12 nucleotide with only G out, we should also get this band. If it is A plus G, we are getting the band, right? That means if that's why we are not getting any band in G, but in AG, that means the uncommon thing here is A, so it will be A. After that, same thing occurs, we get a band in T plus C, but not in C. That means the sequence is T. Again, we get a band in T plus C but not in C, so again it will be T. Then again we are getting a band in T plus C as well as in C. That means the sequence is C. So remember, the easiest way to understand is that if you get bands, same bands, both in T plus C and C, in that case the common one is the sequence. If you are getting only one band for A plus G but not in A or not in G, in that case the uncommon one is the sequence. So that's how you need to understand. For example, again here we get the sequence for A plus G but not for G. That means the sequence definitely is A. Definitely is A, because you're not getting any in the G, right? Right. Now again, if you look at here, we are getting sequence in G as well as A plus G. That means it will be G. So on, we can actually get the sequence from it. So we can see here the sequence C, C, G, A, T, T. So actually we are getting the sequence at the end. So that's how Maxam-Gilbert sequencing actually work, right? So compare it with A plus G with G. If we get band in both, the sequence definitely is the common one, which is G. If we compare it, if we find it only for A but not for A plus G, let's say if we're getting the sequence for say A plus G but not for G, in that case the sequence will be A. So this is the actual funda. If sequence is present in both, the common one is the sequence. If the band is present only in the double one but not in the single one, the uncommon one is the sequence. Okay, so that's the Maxam-Gilbert process. This kind of a little bit complicated, but if you understand this and go back, you can rewind this video and watch it. It will be pretty easy. And this whole picture is taken from the Biochemistry 2nd edition from Matthews and Van Holt, the Benjamin Cummings publication. The copyright goes to the actual producers and developers of this content. It is extremely helpful and a beautiful picture to illustrate. So that's it guys, and I hope that's helpful. Thank you.